Okay, it's 10:30 and we can get started. I have a few housekeeping things too. So we have a few minutes before the actual presentation, but I'll just start talking as people come in. So I want to welcome everyone to the 49th Great Lakes Phragmites Collaborative webinar. I'm Teresa Greninger and I'm joined by my colleague Sam Tank, and we're from the Great Lakes Commission. And we're hosting today's webinar on behalf of the Great Lakes Phragmites Collaborative. Our presenter today is Dr. Gong from the U.S. Army Engineer Research and Development Center, and he will be presenting gene silencing technologies for invasive Phragmites control. But before we hear all about his work and get to the research presentation, I have a few housekeeping items I would like to review. Slide. So for those who may not be familiar, I wanted to introduce the Great Lakes Phragmites Collaborative. The Great Lakes Phragmites Collaborative is a partnership to link people, information, and action on invasive Phragmites. The collaborative is coordinated jointly by the Great Lakes Commission and USGS Great Lakes Science Center with funding from the Great Lakes Restoration Initiative. The collaboration facilitates communication among stakeholders across the region and assists in the development of management tools and programs, and serves as a resource center for information on Phragmites biology, management, and research. The collaborative is led by the Multi-Agency Advisory Committee and applies principles of collective impact to advance the common agenda for Phragmites management in the Great Lakes region. The Great Lakes Phragmites Collaborative is working towards advancing member strategies that are outlined in our common agenda, which you can see here on the screen. The common agenda is the big step towards aligning our regional goals and establishing a shared vision for change. The purpose of the common agenda is to clearly define the intentions for the collaborative, develop a shared understanding of gaps and problem areas surrounding Phragmites, and highlight the areas and strategies that we are working on or wish to work on. So you can learn more about our regional efforts and how you can get involved by visiting our website. And a big component of our work and one way we engage with Phragmites managers is through the Phragmites Adaptive Management Framework, or PAMF. PAMF utilizes participatory science to provide data-driven Phragmites management guidance through an adaptive management framework. So basically this means we have established a way to learn what Phragmites managers are doing and turn that data into annual guidance to help managers make effective and efficient management decisions. So we encourage anyone currently listening, if you're managing Phragmites in the Great Lakes region, to consider joining PAMF. You can learn more by going to the collaborative website, which is listed here on the slide. And if you are interested in joining PAMF, we advise you to take advantage of all of our guidance, and you can enroll in our free virtual training course by following the URL on the screen. We have lots of training resources, and we actually have, I should have put this on the slide, but we have a live training coming up in May. So go to our website and you can register for the live training or any of the virtual trainings. If you have any questions about how to get started or about the program in general, we encourage you to email us at pamf@glc.org. If you're interested in learning more about Phragmites management and research, you should also check out the resources on our website. There's a ton of resources which include management guidance, and of course we have recordings of our past webinars. You can also join our listserv, which connects hundreds of other professionals, and sign up for our bi-weekly newsletter, which includes quarterly research roundup of academic papers published in the previous months. You can join our listserv and sign up for the newsletter on the front page of our website by going to www.greatlakesphragmites.net. Additionally, today's webinar will be recorded and uploaded to our YouTube channel and website, so you can access this webinar and any of our past webinars on our website there. So follow the URL listed, and of course, connect with us on social media and tag us in your Twitter, Instagram, and Facebook post at GLPrag. Okay, quickly, I'd like to cover a few logistical items before we get to the presentation. In order to minimize potential interruptions and background noise, we automatically have muted all attendees, and attendees will stay muted throughout the remaining webinar. To save bandwidth, we ask all attendees keep their cameras off, of course, for the presenter and speaker. And we do encourage written questions from attendees during the webinar. So if you have a question, you may type it in the chat box, which is signified here with a little chat bubble on the webinar screen. And questions may be submitted anytime during the webinar, but your questions will be asked during the end at our Q&A portion. And we'll do our best to take all questions and comments during the webinar. But if you need to leave early, before we've gotten to your question, we encourage you to review the webinar recording that we posted on our website to hear your question answered. And additionally, if you are having any technical difficulties, you may send a message through the chat box, as well as you can direct that message to the host. Or if you want to email us at phragmites@glc.org, someone will get back to you. Okay, I think that's been enough talking from me. So now it's my pleasure to introduce today's presenter. Our presenter today is Dr. Gong, and he is a senior research biologist and principal investigator with the Environmental Laboratory of U.S. Army Engineer Research and Development Center in Vicksburg, Mississippi. He specializes in environmental genomics, bioinformatics, and bioengineering. As a PI, he initiated and led to completion more than 20 multi-year, multidisciplinary R&D projects for both military and civil work customers. He has published over 100 peer-reviewed articles and book chapters and developed more than 8 bioinformatics and computational biology tools. He served on the board of directors of the Mid-South Computational Biology and Bioinformatics Society, as well as editorial board of several prestigious professional journals, including Frontiers in Genetics, Frontiers in Plant Science, Frontiers in Bioengineering and Biotechnology, Experimental Biology, Medicine, and Environmental Toxicology and Chemistry. He's actively involved in educational reach out by holding adjunct faculty positions in several universities, which allows him to co-advise graduate students with academic collaborators. His recent research interests focus on the development of novel biotechnologies for management detection of invasive species, such as non-indigenous common reed Phragmites australis and harmful algal bloom causing cyanobacteria. So thank you so much for sharing your work today, and you can take it away.
Thank you, Teresa.
Yes, you should be able to share your screen over my screen. So.
Okay. Let me share my screen now. All right. Are you able to see my screen?
Yes, I can. Yep, and it's in presentation mode, so it looks great.
Okay, let me turn on my laser pointer. I really love it. Okay, thank you again, Teresa, for your kind introduction. I very much appreciate this opportunity, and I'm so honored to give this talk to the GLPC community. Before I give the presentation, I'd like to first acknowledge my colleagues and collaborators. I've been so fortunate to work with these extraordinary individuals. Particularly, I want to thank Kurt Kowalski, who is in the audience from USGS, who introduced me to the Phragmites community, and Dr. Ed Golenberg of Wayne State University, who pioneered the Phragmites gene silencing work. Without them and other members in our consortium, I won't be able to give this talk. Today's topic may be a little tech savvy for some in the audience. So I'd like to begin with the basics of gene silencing phenomenon. Then I'll give a brief historical review of the large gene silencing technology, or GST family. After that, I'll move on to the centerpiece of today's talk. That is, how do we apply GSTs to Phragmites control? I'll try to answer why we do it, what barriers and solutions there are, and where we are right now. I'll end my talk with an upbeat message about the future perspectives of gene silencing-based next-generation bioherbicides. Gene silencing is a molecular phenomenon that may occur at both transcriptional and post-transcriptional levels. In genetics and molecular biology, transcription refers to the process of copying genetic information from DNA to RNA. So gene transcription can be inactivated by epigenetic events like DNA methylation and histone modification. The transcribed messenger RNA or mRNA can also be degraded by a variety of RNA interference mechanisms, leading to the suppression of gene expression. Epigenetics-based gene silencing has been explored primarily in synthetic biology and biomedical research. Some promising technologies like CRISPR-Cas9-based epigenetic memory writer and memory eraser on the left-hand side, and synthetic epigenetic read-write circuit on the right can program the expression of specific target genes by turning on or off their DNA methylation status in vitro. So far, they're not yet ready for any in vivo gene silencing applications. In today's talk, I'm going to focus on RNAi-based gene silencing technologies. These technologies do not change gene expression or sequence in the target organism. There are two classes of gene silencing agents, or GSAs, worth particular attention because they have been intensively investigated. One is the RNase H-dependent antisense oligos, or ASOs, that can hybridize to the target mRNA, which is then cleaved by the recruited RNase H enzyme. The other is the small interfering RNA, or siRNA, that can also hybridize to the target mRNA, which is then degraded by an enzymatic complex called RNA-induced silencing complex, or RISC. Mother Nature has created many endogenous RNAi pathways for us to explore and build GSTs around them. Let's take eukaryotes as an example. There exist various pathways that can turn double-stranded RNA, or dsRNA, and precursors of hairpin-structured microRNA, or miRNA, and transacting small interfering RNA, or tRNA, into siRNA or miRNA. These mature siRNAs and miRNAs can hybridize to their target mRNA and lead to their cleavage. I believe a lot of you have heard about gene knockout and gene knockdown techniques. These techniques were originally developed for gene function characterization in academic labs. One of the earliest applications of GSTs is RNA-based gene therapy for a wide range of human diseases that are associated with defective genes. For instance, an RNAi drug called patisiran received FDA and EU approval in 2018 to treat a rare genetic disorder called transthyretin amyloidosis, which is caused by a mutation in the transthyretin gene. Here I want to make a clarification that I have no intention nor time to give an exclusive review of all GST applications. Instead, I'd focus on current applications on the control of plant or crop pests and pathogens. RNAi agents can be synthesized either by genetically modified host plants or by other means like in vitro, microbial, and chemical synthesis. Although the so-called host-induced gene silencing approach is very popular in agriculture to protect crops from pests and pathogens, it's obviously not suitable for invasive species control because we don't want to and also don't need to genetically modify the invasive species of concern. So we are most interested in the sprayable RNAi-based strategy. I want to tell you a couple of success stories of sprayable RNAi-based biopesticides. The first story is about a commercial product called Calantha, a Greek name meaning beautiful flower, with an active ingredient called lead-prona. Lead-prona can effectively control Colorado potato beetles via a new mode of action that is RNAi-mediated targeted suppressors. This new MOA was recently approved as Group 35 by the International Insecticide Resistant Action Committee. The CPB is a worldwide pest notorious for its deadly destructive impact on potato, eggplant, and other solanaceous vegetables. Calantha is expected to register in the US this year and be used to control the potato bugs. The second story is about a form called RNA-isense egg, which has also developed proprietary sprayable double-stranded RNA-based insecticides to control Lepidoptera and other insects, pests, for which other RNAi insecticides have low efficacy. Their product is currently in early field trials. One more thing that caught my attention is that their manufacturing pipeline can produce dsRNA at a commercial scale of $1 to $2 per gram. And this opens the door for real-world field-scale applications. Now that we've locked ourselves in sprayable RNAi-based GSTs, I also want to show you a few more laboratory studies that were summarized recently by Rank and Koch. Their review identified many studies demonstrating that dsRNA-derived siRNAs can treat a wide range of plant viruses like sugarcane mosaic virus, cucumber mosaic virus, and the tomato yellow leaf curl virus. There are also multiple studies showing that even naked dsRNA without any protection can effectively cause nearly 100% mortality of target pests in less than 1 week. Again, both naked and nanosheet protected dsRNA can inhibit the growth of many pathogenic fungal species. Now here comes the most interesting part of the review paper. Without genetic engineering the target plant, dsRNA can be delivered in vivo with or without a nanocarrier to Nicotiana benthamiana, which I would call benthy later, to inhibit the expression of both exogenous GFP gene and endogenous magnesium chelatase H gene. These studies provide direct evidence that supports the use of gene silencing to change plant traits and control plant growth. I want to share with you one more paper that just came out in the Plant Journal. The authors used an agro-infiltration method to deliver artificial microRNA or synthetic tRNA expressing cassettes to the benthy leaves. Both types of RNAi agents suppressed the same benthy mRNA coding for magnesium chelators, leading to the bleaching or chlorosis in treated plants. The authors reported significant changes in visible phenotype, chlorophyll A content, artificial microRNA accumulation, and target gene expression. Here, GUS served as the control. More importantly, the authors reported not only local silencing, labeled as LS here, but also systemic silencing, or SS, in the upper non-agro-infiltrated leaves. These results give us more courage and confidence to harness RNAi pathways for invasive plant control. I have to admit that the introduction is a little bit long. Now let's get back to today's topic and talk about the invasive Phragmites control. Currently, we rely on chemical, hydrological, and mechanical methods, as well as prescribed burning to control Phragmites. These technologies have serious limitations and drawbacks, such as resource-intensive, lack of target specificity, residual contamination, and damage to the natural habitat. So there's an urgent need for innovative tools to revolutionize our management toolbox. As I alluded earlier, GSTs, particularly RNAi-based GSTs, may provide an innovative, species-specific, and environmentally benign solution to invasive plant control. A generalized technical approach consists of multiple steps, as illustrated in this slide. It starts with target gene selection, followed by GSA design, modification, and delivery. The GSAs should be screened and optimized through an iterative process in which greenhouse experiments are performed to measure both gene expression and phenotypic changes as a result of GSA treatment. Once the optimal GSAs are identified, we can move on to field trials after we have acquired appropriate permits. In field trials, we should also evaluate GSA impacts on non-target species. Now you may ask, what are the major technical barriers and how do you overcome them? To develop an effective GST, we need to assemble at least 4 essential components: GSA design, GSA modification, GSA delivery vehicle, and GSA application method. Each component constitutes an individual challenge with multiple potential solutions. The most challenging component is obviously GSA design, and the least challenging one is application method. Next, I'll walk you through all 4 components, but I'll do it from the bottom up. For application method, we have seen spray, trunk injection, and irrigation reported in the literature. And the foliar or aerial spray is obviously the preferred one, especially for field applications. As for GSA delivery vehicle, even though naked GSAs have been shown effective in vitro studies, most in vivo studies used one of the nanocarriers, virus, or agrobacterium to deliver GSAs. I want to point out that choice of a delivery vehicle is dependent on the type of GSAs and target plants. For instance, agroinfiltration may be less efficient when used on monocots than on dicots. Phragmites is a monocot, so we should be cautious when we transfect Phragmites with the disarmed Agrobacterium tumefaciens. Why do GSAs need a nanocarrier? The answer is twofold. First, GSAs are negatively charged nucleic acids, which are unable to enter the also negatively charged cell membrane. Nanocarriers like CPPs are positively charged and can neutralize GSAs and deliver the GSAs as a cargo across cell wall and cell membrane via endocytosis. Second, nanocarriers can protect GSAs from enzymatic degradation when they are conjugated with GSAs. Here we show one example of fusion CPPs used to deliver GFP expressing plasmid to plant leaves in vivo. Green fluorescence was observed in treated benthic leaves, demonstrating the expression of GFP. The second example is CNT-based gene delivery system. Single-walled carbon nanotubes functionalized with PEI, CS, or IM have been demonstrated to deliver in vivo plasmid DNA to benthic leaf cells. The infiltrated leaves displayed significantly increased GFP mRNA expression and GFP fluorescence intensity than the controls. The third example is CND-based gene delivery system. Here, a 22 nucleotide siRNA was conjugated with CND and sprayed on benthic leaves. Treated leaves exhibited suppressed expression of an endogenous magnesium chelatase H gene, as well as visible bleaching phenotype for up to 2 weeks post-treatment. These results are similar to those from the agroinfiltration experiment that I showed earlier with artificial microRNA and synthetic tRNA treatments. The only difference is that no systemic effects were observed in this study. The last example of gene delivery system is the well-established virus-induced gene silencing vector. A virus can be engineered to carry a GSA cargo, then the viral vector is transformed into disarmed agrobacterium, which is used to inoculate target plants. In this example, sugarcane mosaic virus was cloned with GSA constructs targeting 2 maize genes, JIH1 and CYP94B1. The knockdown of either or both genes caused the accumulation of jasmonate isoleucine, or JAILE, leading to enhanced pest resistance as reflected as reduced 4 armyworm caterpillar weight. Now let's move up to the next challenge, GSA modification. GSA modification is necessary to either maintain a sustainable supply of GSA or enhance GSA stability and resistance to enzymatic lysis. Internalized GSA expressing cassettes make intracellular GSA synthesis self-sustainable. Earlier, I showed several examples of GFP, artificial microRNA, and synthetic tRNA expressing plasmids. They were all expressed and effectively knocked down their target genes in benthic and other model plant species, leading to the expected phenotypic changes. Here is one more slide on GSA modification. Phosphoryl or PS modification and 2-deoxy-2-fluoro-beta-D-arachnonucleic acid or FANA modification make ASOs resistant to endonuclease degradation. FANA ASO is also capable of self-delivery. I want to say a few more words about FANA ASO technology. It represents the latest breakthrough in the field of GSTs with lots of great features such as self-delivery, excellent cellular uptake, high specificity and affinity, and high stability and resistance. This technology has been patented by AUM Biotech. Recently, 2 published papers reported the efficacy of FANA ASOs in treating a citrus pest vector and a bacteria pathogen. 2 months ago, news broke out that the DoD's Defense Advanced Research Projects Agency, or DARPA, funded a project to develop FANA ASO-mediated GST for the control of the herbicide-resistant pigweed, Homer amaranth. We're also in talk with the AUM Biotech to set up a partnership to develop a similar FANA ASO-based GST for Phragmites control. As I mentioned earlier, there are many different classes of GSAs. Each class acts through a distinct mechanism. The diagram on the bottom right compares the mode of action for the 2 classes of GSAs that we have intensively worked on, that is ASO and siRNA. The key difference is the recruitment of different enzymes used for cleavage of target mRNA, that is RNase H for mRNA ASO hybrids versus RISC for mRNA siRNA hybrids. Now you may ask, what have you done with the Phragmites and how far are you from field trials? So in the interest of time and also to avoid premature disclosure of potentially patentable information, I'm going to briefly summarize the progress we've made in the past 5 years. This slide shows the earliest work we carried out to demonstrate that CPPs can be used as an in vivo vehicle to deliver GSAs to Phragmites leaves. These pictures show how we prepared leaf cross-section slides and observed internalization of FITS-labeled CPP without any GSA cargo. Then we prepared a CPP GSA complex in which the GSA, instead of the CPP, was fluorescence labeled. We used a confocal microscope and observed the fluorescence signal of internalized GSA 4 days post-treatment. The signal intensity was dependent on the ratio between GSA and CPP. A non-functional mock GSA was used in this experiment. We further replaced the labeled mock GSA with the GFP expressing plasmid called PMOD. 10 days after treatment, GFP signal was observed in the CPP plus PMOD treated leaves, but not in the other 2 control treatments, suggesting that PMOD penetrated Phragmites leaves with the help of CPP and expressed the GFP construct in leaf cells in vivo. We then performed a series of greenhouse experiments to test GSA nanocarrier combinations on Phragmites. The GSAs were designed to target select Phragmites-specific essential genes identified from the Phragmites genome, which was recently published by our consortium. The genome sequencing and annotation effort was led by Kurt Kowalski and funded by GLRI. What's shown here are some of our greenhouse experimental results. Besides CPP, we also used CNT and CND as the delivery vehicle. We tested both dsRNA and PSASO designed to target two PDS gene isoforms in Phragmites, PDS 3.1, PDS 3.2. Our results indicate significant inhibition of target gene expression 3 or 4 days post-treatment for all the GSA nanocarrier combinations. However, no phenotypic changes, that is, bleaching or chlorosis, were observed, suggesting that the regulation of the expected phenotype may involve other unknown genes. We also used the CPP to deliver the dsRNA targeting the two Phragmites PDS genes. However, we only observed marginally significant inhibition of PDS 3.1 on day 7. In comparison with the results for CND and CNT shown in the previous slide, we may conclude that CPP is probably less efficient than CND and CNT. We also tested the fauna ASOs on the benthic plants. 4 fauna ASOs were designed to target the benthic PDS gene. All except one induced significant target gene inhibition on day 7, but only fauna ASO4 suppressed PDS gene expression on both day 7 and day 10. No visible phenotypic change was observed in either local treated leaves or systemic upper leaves throughout the experiment. These results suggest that we may need to design more potent fauna ASOs and increase the concentration of applied fauna ASOs. This slide shows our latest effort. In collaboration with Professor Steve Witham of Iowa State University and Professor Danish Kumar of UC Davis, we launched this new effort to develop VIX vectors. The first step was to identify a virus that can infect Phragmites both locally and systemically. Shown on the left are the gel pictures from RT-PCR of the BMV code protein and BSMV gamma B protein. These results suggest that both BMV and BSMV can not only replicate in the inoculated Phragmites leaves, but also move systemically to upper non-inoculated leaves, even though at low 1 or 2 out of 6 odds. Our next step is to make GSA constructs and clone them into these viral genomes. Then we inoculate Phragmites and verify if the GSAs can be expressed and inhibit target genes both locally and systemically. Meanwhile, we're also looking into how to improve the systemic movement efficiency. Now, to summarize, there are undoubtedly lots of challenges and opportunities in applying GSTs to invasive Phragmites control. Much remains to be done, and there are many unresolved issues. We've screened dozens of GSA nanocarrier combinations, but have not yet found the optimal one. We haven't seen the desired phenotypic changes. Even the target gene expression was knocked down. To induce systemic silencing, we may have to use the VIX approach. And I anticipate that our final product is likely going to target multiple genes via different pathways. In view of the success of RNAi-based biopesticides, I'm optimistic and confident that we should be able to discover RNAi-based sprayable bioherbicides to mitigate the invasive Phragmites and other invasive diseases. Among all the challenges we face, identification and characterization of target genes and their silencing pathways are critically important. We have tested different classes of GSAs and delivery vehicles, including nanocarriers, virus, and self-delivering fauna ASOs. It is our belief that we should assemble these components in a manner to enable synergistic induction of gene silencing and irreversible phenotypic changes leading to growth inhibition. I want to conclude my talk with this optimistic future perspective. It's a quote from the same review paper by Rank and Koch that I cited in my earlier slides, but I replaced pest with herb. So now it reads, the sudden rise in demand for herbicide alternatives has boosted research on sprayable RNAi bioherbicides, generating significant technological developments and advancing the potential for field applications in the near future. I hope I can come back next year to give another talk with more convincing results that would allow us to launch field trials. I think I used about 40 minutes to walk you through a 5-year-long journey. Along this journey, more and more individuals and institutions joined us. Thanks to the continuous funding and support we received over the course, particularly from GLRI, both our efforts and our consortium have expanded way beyond our initial vision. If you're interested in joining us and being part of the endeavor, please contact me. I believe Teresa also has my contact info. Thank you all for taking your time and being with us. I think that's all I have. Now I may take your questions.
Thank you so much. Yes, let's.
I want to exit mode.
There. Okay.
Okay.
Thank you so much for that amazing presentation. I agree, you packed a lot of work in 40 minutes, and that was very impressive. And I also hope you can give an update in a few years with like where this research is going. It's very exciting.
Yeah, I hope so too.
But yeah, so thank you. If you guys see your screen, here's his contact information if you want to reach out if you have any questions. And right now we can take questions from the audience. Please, since you guys are muted, use the chat box, which is the chat bubble, either on the lower part of your screen or upper part of your screen, depends on what app you're using. So we do have a question. What is the time? Excuse me, let me read this. What is the timeline for widespread application of this technology, and what impacts do you foresee having on Phragmites management?
Okay, the timeline.
Kind of broad.
Well, yeah, that's a good question. I wish tomorrow, but realistically, we are looking at a couple of years to roll out an effective product. Since we got the some additional funding from GRI, which would boost and access the the research and development effort. So I'm looking to introducing more folks into our consortium and get more help. And also that's why we started the collaboration with the academics to develop VIX vectors. So that's part of our new effort. And so yeah, a couple of years probably it's reasonable. And as far as impact on management, if the product can be affordable, like I mentioned, $1 or $2 per gram, I think it'll have a huge impact on the management, because right now I don't have the numbers, but I think it costs millions of dollars to right now to control the Phragmites. And if we can invest on the use of the next generation herbicides, I think everybody would agree to go for that, because it's environmentally benign, and species specific, and they have lots of advantages over the current technologies. I hope I answered your question.
You did, yes. We have another question kind of related to cost, so I'll weave that in. How much, so I guess when this product is eventually available, do you envision it, the cost being comparable to that of herbicide products being used, or potentially cheaper?
Yeah, it depends on the scale, I believe. If this becomes a household product, I believe the price would go down dramatically. So it should be affordable and comparable.
Are there similar processes for approval of this technology as would be for the release of biocontrol agents? That question's from Francine.
The process, you mean the regulatory approval?
Yes.
I'm not quite sure. Probably Kurt can help answer that question. Kurt Kowalski, he has done some work on that front. Kurt, are you still there?
Yeah, I am. Yeah, I am. Thanks, Dave. Hopefully you can hear me okay.
Yeah, we can.
Good question, Francine. Yeah, at this point, we understand that it would be regulated. Technology like this would be regulated by EPA more as a bioherbicide than as a biocontrol agent. And we haven't gone through that process yet, but just in the discussions that we've had already, we do believe there is a regulatory pathway to registration and to move it forward once the technology gets worked out through time.
Thank you, Kurt. I hope that answered the Francine's question.
Okay, we have a question from Julia. Do you know if there's any other work of similar technologies for invasive typha, so cattails, or what others maybe invasive species are being targeted for this?
Yeah, the only other effort we are aware of is the one that I mentioned in my talk, DARPA-funded effort on pigweed, the invasive pigweed.
Okay. A question from Mark. Does this research also include work to show that potential effects to non-target species is low / non-existent, hopefully?
Could you repeat that question?
Yep.
I didn't get.
Does this research, will this research also include work to show that that there are no potential effects to non-target species and that are low or non-existent?
Yeah, potentially. Well, our goal is to make species specific. So it shouldn't affect the non-target species in theory. However, before you do the real field test, there's still some chances that it could hit non-target species because of the conservation of the target genes. Some of the target genes are very conserved, across species. So, but when we designed the GSAs, we took that into consideration. But however, we don't know all the gene sequences of all the species, plant species out in the field. So we still need to run a field testing to see if there are any non-target impact.
Okay. If anyone has any other questions, add them now. Right now, it looks like you've done a great job answering them all. So, and I would reiterate that if you think of a question later on, maybe later today, sometime next week, feel free to contact him. Here's his contact information. I'm sure he'd be more than happy to answer your questions. And so this webinar today will be recorded and posted on our website. So if you want to view it again or share it out with colleagues, check back on our website in a few days. It will also be sent out through the Eventbrite. If that's registration, we'll send it out there too. And this is our last webinar for a few months, at least for this spring summer series. So thank you, Dr. Gong, for wrapping us up. If anyone is interested in the PAMP virtual training session, that will be May 25th, and you can go to our Great Lakes Phragmites website and it will be on there, or email us. We would be happy to help you get registered. And we want to acknowledge the U.S. Geological Survey and the Great Lakes Restoration Initiative for providing funding and support for our collaborative. And as a reminder, go back to our website to find the webinar recording in the next few days. And if you have any additional follow-up questions or comments, please feel free to email us at phragmites@glc.org. And so thank you again for presenting with us, Dr. Gong, and thank you everyone for joining us.
Thank you. Thank you for the opportunity.
Yes.
Great pleasure.